Now let's start with the next concept in this chapter, that is heredity. Right? Now, what do you mean by heredity? Till now we've discussed accumulation of variations, right? And now let's look into the next concept, that is heredity. Now, heredity basically refers to the transfer of characteristics, okay? Transfer of characteristics from parents to offsprings. From parents to offsprings. So when characteristics are transferred from parents to offsprings, we call that heredity. Transfer of characteristics, right? That process of transfer is called heredity. Now we've heard this many times. There is a hereditary king, there is a hereditary ruler, there's a hereditary master. So these basically mean that that person is a hereditary ruler. He's going to get that particular ruling ability, ruling rights from his parents, right? So similarly in biology, we refer to heredity as transfer of characteristics, genetic characteristics from parents to offsprings, right? That is your heredity. Now, we study heredity and it gives us an idea about genetics, heredity and inheritance. There's another concept called inheritance. Okay. Heredity and inheritance together they form the basis of genetics. Okay. Together they form the basis, the main basis of genetics. That is called heredity and inheritance. Together they form genetics. Now, the father of genetics and father of heredity is Gregor Mendel. Gregor Mendel. And Gregor Mendel was basically a monk. Okay. He was a monk and he used to experiment with organisms. He was very fascinated with expression of traits, with the expression of particular characteristics. Okay. And he basically studied this transfer of characteristics and this expression of characteristics in offsprings from parents, right? And he basically conducted some experiments, right? He conducted experiments and these were called Mendelian experiments. Mendelian experiments. Now, what were these Mendelian experiments? He basically took pea plants. Pea plants, okay? And their biological name or scientific name is Pisum sativum. So he basically took pea plants, that is Pisum sativum, and he conducted, reproductive research. He crossed different pea plants with different traits and he studied the characteristics and traits which were expressed in the offspring, right? So that was his study. And he studied 7 different traits. 7 different traits. Now, what were these traits? Well, let's just list them down. There was height of the plant. Okay, he studied the height of the plant after offspring and the parent generation. He crossed them, then whatever the offspring, he studied the height of the offspring. Okay, that he did. After that, he also studied the shape of the seed. Okay, he studied the shape of the seed of the plant, right? He studied the color of the seeds. Okay, then he studied the color of flowers. Color of flowers. Then he studied the stem structure. Okay, he studied the stem structure. These are 5, right? Then he studied leaf arrangement. Right? So he studied these characteristics. These are 6. There was one more. but I don't seem to really remember that. But these were some of which he studied, 6 traits. Out of this, in your 10th standard course, there is only height of plant, shape of seed, and color of flowers. Right? And that too, this color of flowers is very, very minor. Mainly you have these three: height of plant, shape of seed, and color of seed, right? Height of plant you will study, shape of seed you will study, and color of seed you will study, right? And he performed many kinds of crosses, right? He performed a monohybrid cross, right? Then he performed dihybrid cross. He also performed trihybrid cross. He also performed tetrahybrid cross. He also performed penta, but for now just leave this. Right, so he performed many types of crosses, right? This is cross basically means cross pollination. So between two plants. So monohybrid cross means mono means one, right? Mono means one. So a cross between plants with one pair of contrasting characteristics. One pair of contrasting characteristics. So when he crossed two plants which had one pair of contrasting characteristics, where he studied one pair of contrasting characteristics only, then that was called a monohybrid cross. He crossed two plants and studied one pair of contrasting characteristics. It could be the height of those plants, it could be the shape of the shape of the seeds of those plants, it could be the color of those plants, and etc, etc. Okay, so one pair of contrasting characteristics. In dihybrid, he studied two pairs. So for example, it could be he's studying shape of the seed along with the color of the seed. He's studying shape of the flower along with color of the flower. He's studying shape of the stem along with color of the flower. So he's studying two characteristics together, right? And that is dihybrid cross. And trihybrid and tetra, this is 3 and this is 4, right? In your again 10 standard course, you only have mono and dihybrid crosses. You don't have trihybrid and tetrahybrid. You only have monohybrid and dihybrid, right? So these are the two crosses we are going to study in Mendel's crosses, right? And he did these crosses. He studied the different characteristics which came about. He studied genotype and whatnot, right? And then he came to the conclusion, okay, he came to the conclusion of what? He concluded with some laws, right? He came to some laws and he generated the laws of inheritance. The laws of inheritance. Okay, he studied the laws of inheritance. He formulated laws of inheritance. He formulated 3 laws of inheritance. First is law of dominance. Then second is law of segregation. And lastly, we have the law of independent assortment. Okay, law of independent assortment, right? So these are some 3 laws of inheritance which he formulated as a result of his crosses which he performed and the traits that he studied, right? So I hope that's absolutely clear. This was an introduction to Mendelian experiments, how Mendel performed his experiments, what all he studied, how he studied them, and what he came to conclusions to, right? Now, before we conclude this small video, I want to just go over some concepts, right? Now, when Mendel studied these things, right, he came to some conclusions. For example, there is a particular trait, for example, height, right? Height. Now, height is denoted by a particular gene, right? You know what a gene is now. I have explained to you what a gene is. Now, gene is a functional unit of DNA. And a particular gene which an organism obtains from its parents, okay, they form combinations. Now, for example, we have the mother and then we have the father. Now, the mother as suppose, because she is going to give the egg, right? She's going to be the source of the egg, and the father is going to be the source of the sperm. Now, the mother, it is to be noted here, whenever there is formation of gametes, right? It occurs through a kind of cell division which is called meiosis. Because there are certain germ cells, certain cells present in the reproductive organs which undergo a kind of cell division called meiosis, and they form these cells. To know more about meiosis, you can look up my series on cell division. Now, mother and father, they're going to give their reproductive gametes, reproductive cells. And what is going to happen is that each of these gametes is going to carry one particular gene form. One particular gene form for a particular trait, right? So this mother may carry some, for example, we are talking about some trait, some random trait, suppose height. So this mother may carry a gene form for height as capital T. And this father may carry another gene form of height, uh, and it might carry a gene form of height as small t, right? So basically there are gene forms, right? There are forms of a gene of a particular trait. For example, height, it can have two types of gene forms. It can be capital T and it can be small t. Capital T is the tall form, tall gene form. And then small t is the short gene form, short gene form. Now capital T is the tall gene form and short gene form is small t, right? So when these recombine, they will give rise to a zygote which will have a combination of this in terms of its height. And this will form a gene form pair, right? This will form a pair of gene forms, right? And this pair of gene forms is going to constitute the genotype of the organism, of the progeny. So what is basically genotype? Genotype is basically the genetic constitution of a particular organism with respect to a particular trait. Okay, it is the genetic constitution. Genotype is basically the genetic constitution. Okay, the genetic constitution of an organism, okay, with respect to a particular trait, right? That is called the genotype. So here it got one this capital T gene form and small t gene form, and got capital T small t as its genotype, right? These gene forms, okay, these forms of a gene which we get, they are called alleles. They are called alleles, and alleles are basically gene forms or forms of a gene. So here for the gene for height, these are the two forms you may get, right? So basically alleles form pairs and form genotypes. Okay, there are allelic pairs. This is called an allelic pair. And these allelic pairs are what form the genotype of the progeny or of an organism, right? Now, one allele is present in the sperm and one in the egg, right? For a particular trait. Now, there may be different alleles, right? So, each reproductive cell has one allele, and when these two fuse together, they form an allelic pair. And depending upon the allelic pair and the genotype produced because of it, there will be expression of the trait. There will be expression of the trait, and the expression of this trait which is visible in the progeny is called the phenotype. Okay, it is called the phenotype. Phenotype is basically the visible expression of a trait or visible characteristics of an organism. Visible characters of an organism. So the visible characters which can be seen in an organism, due to its genotype is what is called the visible characteristic of an organism, and that is called the phenotype. Phenotype is the external characteristics which are visible in a particular organism. They form its phenotype. And phenotype is subject to the genotype. Whatever the genotype may be, depending on that, you will have a phenotype, suitable phenotype, right? Now, in this case, this capital T is a dominant trait and small t is a recessive trait, and together they form capital T, small t. So this dominant trait will suppress the recessive trait and you will get a tall progeny. You will understand recessive and dominant when I talk about Mendelian's law of dominance and all that. But for now, just try to understand that this is what happens. This is the basic idea, right? This is how basically phenotypes and genotypes, and you can see different traits and the genetic constitution takes place, right? I hope that's absolutely clear. So for here, we're done with heredity. In the next video, I'll be beginning with Mendelian experiments, and then we'll go on to see how Mendel determined his laws. Thank you very much.